So basically, you all are prepared already. So genetics is something which is very volatile. So my only endeavor is to get you into the groove for genetics for MRCP, number one. Number two, to catch hold of almost everything which is important for the exam. Number three, not to miss out on something which is important. That is also very, important for the exam. So we will just touch upon few topics. We'll just briefly try to understand them because in such a short span of time, whole of the genetics and the diseases associated with it, it becomes very difficult. So as far as the genetics goes, you need to understand that in genetics, you will get number one, the genetic basis of disease. This is the concept that is asked in the exam, number one. Number two, how to diagnose genetic diseases. And number three, which is a very weird thing, it is how to treat genetic diseases. And number four is miscellaneous. Miscellaneous is like pharmacogenetics, then screening for genetic disorders. Like every country will have their own set of recommendations and guidelines as to how to screen for genetic diseases. So just this, with this generic background, I would like to ask randomly, can anybody tell me what is the genetic basis of a disease? Broadly speaking, why it is important for us to understand the genetic basis of a disease? See, some diseases, they are inherited as a genetic disorder. Now, this is one. Number two is certain people are prone to certain diseases because they have genetic deficiencies. For example, G6PD deficiency. This is also a genetic disorder. Like some people who are hemophiliacs, they have genetic disorders, so they're more prone to develop bleeding disorders. Number three, certain cancers, they are more common in certain genetic disorders. Number four, certain structural diseases are more common in certain genetic disorders. So we as doctors, we have the responsibility to ascertain if somebody is carrying a gene or a chromosome which can affect the offspring or their own life in a particularly wrong fashion. Then comes diagnosis. Can anybody tell me how do you diagnose genetic diseases? Am I audible? All I'm asking is, if somebody can tell me how do you diagnose genetic diseases? You are preparing for MRCP exam. You already have certain amount of knowledge because all of you are doctors. So my question to all of you is, how do you diagnose genetic diseases? Anyone? Okay, so you will, yeah.
Screening the genetic deficiency, the genes, what we mentioned.
Yeah, how do we diagnose these genes? What is the mechanism? Like we order sodium, potassium, urea, creatinine, CBC, total blood count, LFT. What do we say that we want to get a genetic disorder tested?
The genetic mapping.
So we do a karyotyping, yes, and gene analysis. This is what is done these days. So you will enjoy this class after having these questions in mind. Then, of course, how do you treat genetic disorders? That is another thing. And then we will see the miscellaneous pharmacology. Miscellaneous pharmacology means certain enzymes, they are inherited and they have different, fast acetylators and slow acetylators. These are all genetics. Okay. So let's begin the topic with the biggest chunk of the genetic disorders, that is autosomal dominant disorders. Now, I always tell my students that there are two types of disorders as far as autosomal genes are concerned, which are autosomal dominant and autosomal recessive. The autosomal dominant diseases are more or less structural diseases. So wherever you will find a structural disease, no, there are high chances that the patient will have a autosomal dominant disorder. Then comes autosomal recessive disorders. Autosomal recessive disorders, they usually manifest as enzymes or chemical related processes deficiency. So they will have like some enzyme deficiencies are usually autosomal. Wherever it is a biochemical process related with it, it usually is autosomal recessive disorder. So let's begin our journey for genetics by autosomal disorders. So all you need is one mutated copy. So we all know that there are two chromosomes. So genes are present on both of them. One is from the father, one is from the mother. So all you need is a major single gene which is altered and it will manifest as a disease. A one mutated copy of a gene is sufficient. For example, A, this is a normal genotype. A, this is female, this is male. They marry, so everybody has A, we understand that. But now let us say the gene is so dominant that this gene became mutated. So we have A dash A and AA. So A dash A will be diseased. That means you will never have a carrier. Never a carrier. Why? Because even a single copy is sufficient for the disease to manifest. Hence, there will be no carriers in autosomal dominant disorder because the genetic defect is a dominant defect. Even if it is singly present, it will definitely cause the disease. Common examples are Huntington's chorea, Marfan's syndrome, familial hypercholesterolemia. So, let us go through the case scenarios to understand the process in a better manner. A 28-year-old man with a family history of sudden cardiac death is diagnosed with aortic root dilatation on echocardiogram. He is tall, he has long fingers, and there is a chest deformity known as pectus excavatum. What is the most likely disorder in this patient? Ehlers-Danlos syndrome, Marfan syndrome, Turner syndrome, or Loeys-Dietz syndrome?
Marfan's.
Yeah. So this is Marfan's syndrome. How do you say that? Why not Ehlers-Danlos?
Cardiac defect, I think, is only for Marfan's.
Say again. Pectus
The cardiac,
excavatum.
the cardiac defect is not seen in Ehlers, I think.
Yeah. So aortic root dilatation, very aptly picked up. So aortic root dilatation is usually seen in this Marfan syndrome. Okay. So if I had to pick up something for Ehlers-Danlos syndrome, then what would it be? Because both of them, they manifest very, very similarly.
The lens subluxation will be
Wonderful. So lenticular subluxation. So if there was mentioned that there is a history of a repeated blurring of vision for which the patient has been diagnosed with some lens related issue, this will be Ehlers-Danlos syndrome. Okay. So this patient has Marfan syndrome. We will come to it that there is a mutation in specific gene known as fibrillin-1 gene, which is FBN1 gene, characterized by, as you said, aortic root disease, tall stature and skeletal abnormality. Tall stature and skeletal abnormality, hyperreflexia, hyperlaxity, these are seen in other connective tissue disorders also, but aortic root dilatation is specific for Marfan syndrome. Okay. Can anybody tell me why Turner syndrome is specifically mentioned as a choice in this question? Now, as a student, you need to read the examiner's mind also. Why Turner syndrome is kept in this? Because
Yeah, they also have some cardiac related issues, is it?
Do they have a tall stature?
No.
Are they not tall? Turner syndrome? Turner is what, XO?
Yeah, 45 XO.
Yeah, so they are usually tall people, but they have aortic root issues. What are those issues they have?
The
So they usually
coarctation
have
of aorta.
coarctation of aorta. Yeah, so they have coarctation of aorta. So when you have similar two things in a question, then there are chances that some student who have partial knowledge, they might pick up that answer. So it is important for you to differentiate. Every single word is important. In MRCP exam, nothing is taken for granted. All right. So the answer here is Marfan syndrome.